Hi guys, most of the pracs that we have been filming have been pracs that you really couldn't do at home without those prepared slides, without actually, making the slides up and actually having something that you can actually film on the microscope really. And most of you don't have microscopes at home. But this one you can do at home, well, at least part of it. Now what all you have to do is go out into nature. Ooh, nature, and get yourself a leaf. Where'd I put my leaf? Get yourself a leaf, because this is all you need to do the first part of practical investigation 5.2 to observe leaf structure. Looking at the midrib, the petiole, and the veins, and giving a kind of close look at that and actually doing a diagram, because that's a big part of biology, is actually observation and writing down your observations. Because I don't actually know how many times you guys will watch a movie like Jurassic Park or The Simpsons, and actually think about the fact that there's the leaves there, and actually see that those things are an important part of our world and an important thing for us to think about, because they're the things that are actually leading to our world as we know it, where we have so much oxygen in our atmosphere, and it's like the perfect world for us to live in. And that's because of photosynthesis and because oxygen is being released mostly by the leaves of plants. Like the taiga forest in Siberia, that place has probably the most photosynthesis going on out of anywhere, and actually it's contributing a great percentage of the oxygen atmosphere. So maybe even after this prac you might just have a bit of a enlightenment on how important leaves are. Thanks. Okay, so what we just saw there was Mr. Ellis showing us how to make a microtome slide. So the microtomes, that one technology that we spoke about in the technology PowerPoint. And really what you're doing here is that you've got these two bits of glass here, and you're trying to embed a section of a leaf or a section of what you're trying to put onto a slide. Then what you're doing is you're taking this very sharp, very sharp, it's like an old barber's blade. And what you're doing is you're scraping off the top. So that top layer has been scraped off already, so nothing's going to come off. And you're scraping it along and trying to get a thin section, again, so the light can go through it on the microscope. What we're going to try and do is try and mount these two that are here. So following some very unskilled work, I guess, both a bit out of our depth there, Mr. Me and Mr. Ellis. I haven't done it for a while, and Mr. Ellis hadn't really done it with that particular microtome, but you can see that it can produce a slide where you can start to see some definition, but for our purposes and for our actual looking at observing leaf structure, practical investigation, this is not going to be really that useful for us. But it's good to see that there is a different way of doing it, especially if you can do it properly. You can get good results, and the one I'm going to show you now is actually one that you can use that'll be a bit better for you, well, for us to do this practical. So I'll get that slide up now. Okay, so now we've got one that's done by professionals, this transverse section of a leaf. So this is aligned to, again, investigation 5.2, and this is from in the method step 4 down to step 12. So I'll include the picture that they want you to take at question 12 for this. So it'll just be a screenshot like this. But so now, at least now we can actually see, so if I just make this big, you can actually see the upper epidermis here and the lower epidermis. The reason I can infer, really, other than looking at the vascular bundle here, that this is the lower epidermis, is that there's actually the stomata. So you can actually see those things here, and with the associated cells. What were those two cells called? Those really high specialized cells. What were they called in each stomata? There's the stoma, and then controlling the stoma were those two cells. What were those two cells called? But what we want to actually look at here is the different cells, the different parts of structures that are present here, and probably talk a little bit about their functions as well. So firstly, on this the top layer of the leaf here, we have the cuticle, and the lower level we have the cuticle. The cuticle is helping stop water loss in hot, dry climates. So if again we go to our leaf here, because we're going to be a bit tree huggery in this, you have our leaf here, quite waxy, quite rough feeling, quite like almost leathery feeling. That's that waxy cuticle that's stopping water loss in our hot, dry climate here in Australia. That's why our leaves are staying green for quite a lot of time on the trees there. So then you've also got this our epidermis layer here, lower epidermis layer, upper epidermis. Maybe you're seeing slight changes there in the size of the cells, but that could be just partly on this part of the slide. Then you've got our photosynthetic cells. You can see these are the greener ones that actually have the chloroplast contained in them. So now we've got these chloroplast containing cells, the palisade cells, densely packed together, really stacked really quite evenly along each other. They're really just their maximum photosynthesis synthesis actually occurring. Again, you can see on this leaf here that I've been using, really dark on this side, not as dark on this side. This side's the side that's facing the sun most of the time to try and find the sun. It's kind of like a solar panel looking for the light. It's just trying to get as much of that light energy coming in so that photosynthetic reaction can actually take place. And then our next most powerful sort of photosynthetic cells are the mesophyll cells, our spongy cells here. Not very densely packed, right? So not very densely packed. Again, you can follow on page 140 to look at this. Not very densely packed, but still undergoing photosynthesis, still containing chloroplasts. these are not as densely packed because they're trying to maximize any gas exchange that might be occurring within the leaf by not having not being so confined in terms of in space. And then you've got our strength cells here, either side of the vascular bundle. That's our parenchyma. That's actually providing a bit of strength there. And then you've got our xylem. You can't really see the cambium here, but then the phloem here in our vascular bundle. And you can see there's another beginning of the vascular bundle here. And again, our stomata. And those very highly specialized cells that compose part of them. What were they again? This is aligned to investigation 5.2, a practical investigation to observe leaf structure. The way they get you to do it is to stack two to have the leaf section stacked between two slides. So sorry, I should have done this earlier. Have that leaf stacked in here, and then you cut thin slices off into water, and then you're able to collect those and wet mount them onto a slide. So similar to the microtome, just the microtome trying to achieve a much thinner slice and therefore give you the most clarity. But yeah, there's a little bit more involved in that. And so go ahead and complete the discussion questions and give your results of this investigation, which is looking at this as a leaf whole and then the transverse section where you've got the slide cut off. So again, thanks for engaging with this and thanks for watching.